Hello everyone, I am Deval and we are covering the topic of chapter sexual reproduction in flowering plants. So the topic is pollination. So what is pollination? The process of transfer of pollen grains from anther to the stigma of pistil is known as pollination. Now there are two types of pollination, self-pollination and cross-pollination. Now, self-pollination is divided in two parts, autogamy and geitonogamy, and cross-pollination have a single type known as xenogamy. Now, let's talk about autogamy. Pollination is achieved within the same flower. It means when the pollen grains are transferred from anther to stigma of the same flower, it is known as autogamy. Now, in flowers with exposed anther and stigma, a complete autogamy is rare. If we want, then it needs two conditions. One, anther and stigma should lie close to each other. And second, there should be synchrony in pollen release and stigma receptivity. It means the time of pollen release and stigma receptivity should be same. Now let's see the example of autogamy. These are some examples of autogamy. As you can see, oxyls produce two types of flower. Let's see the two types of flowers. One is chasmogamous. They are similar to flowers of other species with exposed anther and stigma. Example of this is hibiscus, as you can see in picture. Now let's see the second type of this. It is clastrogamous. They do not open at all. Anther and stigma lie close to each other and they are autogamous as there is no chance of cross-pollination. They produce seed set even in the absence of pollinators. It means they are not dependent on pollinators for pollination. Example of this is cadamine. Moving ahead, we got geitonogamy here. Transfer of pollen grain from anther to stigma of another flower of the same plant. It means when the pollen grains are transferred from anther to stigma of another flower of the same plant. It is known as geitonogamy. It involves pollinating agents. Xenogamy. Transfer of pollen grains from anther to the stigma of different plant. It brings genetic difference, means variation. And it's simply we can call it cross-pollination. Moving ahead, we have agents for pollination which are of two types, abiotic and biotic. In abiotic we have wind and water, and in biotic we have insects like bees, moths, wasps and flies. And we also have birds and bats. We'll talk about abiotic agents. In abiotic we have got wind pollination. It's also known as anemophily. Of wind pollinated flowers are lightweight, non-sticky and winged sometimes. Anther are well exposed for easy dispersal of pollen grains. And stigma is often large and feathery to easy trap of airborne pollen grains. Examples are oats and corns. Water pollination, also known as hydrophily. Water pollinated flowers are small and colorless, fragranceless and nectarless because they are less dependent on biotic agents. They got non-vertebral floral parts except pollen grains and stigma. Stigma should be long and sticky. Now water pollination is limited to about 30 genera and mostly seen in monocotyledons. In some of them, the female flowers reach the surface of water by long stalk and male flowers or pollen grains are released on the surface of water. These male flowers or pollen grains are carried by water currents and reach female flowers. An example of these is Vallisneria. In some of them, the female flower remains submerged in water and long ribbon-like pollen grain carried inside water and reach the stigma. The best example for this is seagrass. Some flowers emerged above the level of water and got pollinated by entomophily or anemophily. It means by insect or wind. It mainly seen in Vallisneria and Hydrilla inside fresh water and Zostera in marine seagrass. Pollens are protected from wetting by mucilaginous covering on it, and here you can see some picture of them. Insect pollination. When the pollination takes place by insects, it is known as entomophily. Here are some characteristics of entomophilous flowers. Flowers are large, colorful, fragrant, and rich in nectar. When the flowers are small, they form inflorescences to make them visible. The flower, pollinated by flies and beetles, secrete fall odor to attract these animals. The pollen grains are generally sticky. Some species provide safe places to lay egg. Example, Anelphophilus and Yucca. There is a very close obligatory symbiotic relationship between species of moth and plant Yucca. They cannot complete their life cycles without each other. The moth deposits its egg in locule of the ovary and the flower gets pollinated by moth. The larvae of moth come out of the eggs and the seeds start developing. And there are many insects which consume pollen or nectar without bringing about pollination. They are called pollen or nectar robbers. Bird pollination, also known as ornithophily. Bird pollinated flowers are brightly colored without fragrance and should produce nectar. Should have funnel shaped corolla. Example of this is bombyx. It's also known as red silk cotton. Here you can see picture of that. Outbreeding devices. Devices for promoting cross-pollination. In order to avoid self-pollination, cross-pollination is encouraged in plants as followed. A. Avoiding synchronization. In some species, pollen release and stigma receptivity are not synchronized. The anther and stigma mature at different times, and this phenomena is called as dichogamy. B. Arrangement of anther and stigma at different positions. In some species, the arrangement of anther and stigma at different positions prevent autogamy. C. Self-incompatibility. It prevents pollen of one flower to germinate on the stigma of same flower on the same plant due to presence of similar sterile gene in pollen and stigma both. D. Production of unisexual flowers. It's also known as declining, where the male and female flowers are present on the different plants, prevent both autogamy and gynogamy. The best example for this is pollen pistil interaction. It is a dynamic process involving pollen recognition followed by promotion or inhibition of pollen. This interaction takes place through the chemical components produced by them. If the pollen is compatible, then pistil accept it and promote post-fertilization events. The pollen grains germinate on the stigma to produce a pollen tube through one of the germ pores. The content of pollen grain move into the pollen tube. The pollen tube grow through the tissue of stigma and style and reach the ovary. If the pollen is incompatible, then the pistil rejects pollen by preventing pollen germination on the stigma or pollen tube growth in style. The pollen tube, after reaching the ovary, enters the ovule through the microfilar or chalazal end and then enters one of synergids through the filiform apparatus. The filiform apparatus present at the microfilar part of synergids guide the entry of pollen tube. And here we have some diagram of pollen pistil interaction. Artificial hybridization. It is one of the major approaches of crop improvement by using desired pollen grain for pollination. This is achieved by emasculation and bagging techniques. Emasculation is removal of anthers by using force kept from bisexual flower bud of female parent before the anther dihyles. The emasculated flowers are then covered with a suitable bag made up of butter paper to prevent contamination of its stigma with unwanted pollen. This is called bagging. And here we have some pictures of emasculation and bagging.